In the 1960s, something happened that really changed our entire view of who we were and what the world was. And that was when we landed on the moon. When you see the Earth from that perspective as this little tiny blue island floating in that vast sea of nothingness, it did more to convince us that this was one world than all the diplomatic efforts ever tried. So now there's an equally important revolution happening that's going to change our view of ourselves. But most people aren't really paying very much attention to it, and that's this business of DNA. Because DNA technology is advancing so quickly with such blinding speed that it's going to take us all by surprise. For example, the human genome was sequenced 10 years, billions of dollars. Now, today, you can do an entire human genome in a day for under €1,000, and the price is still coming down. So as the code of life, the source code of life, becomes more and more hackable and life becomes less and less of a black box, it's really going to start changing everything. So who should have access to this technology? I think a lot of people would be pretty comfortable with this fellow, but what about this guy? So GenSpace, as I said, is a new concept. It's this open community laboratory. So we support ourselves with adult education classes, with membership dues for the people that do projects there. It's really a biohacker space. And I can give you 3 good reasons for these things to exist. The first is we really need more public outreach in science. All of this technology comes loaded with social and moral and ethical questions. And I really think people should be coming to the discussion from the viewpoint of someone who understands a little bit, at least, about the technology. I'm very much afraid that it's going to be fear and ignorance that drives policy. The second is that these are fabulous places to innovate. We've hacked software and now hardware, and now we're moving into hacking wetware. So I'm very, very interested in what these spaces are going to produce in the long run. It's just at the beginning, mind you. And the third is that 65% of the jobs that your children will have haven't even been invented yet. And so we do provide an awful lot of support for educational systems in science education, particularly like very, very cutting-edge science stuff. And we think of ourselves as a bridge. We're in Brooklyn, so that's the Brooklyn Bridge. But we think of ourselves between a bridge, between science and the general public, which I think is very much needed. Now, of course, this is the first thing that a lot of people think of when they think of a biohacker space. And I'd just like to tell you that we've spent an awful lot of time really considering this stuff. We have a science advisory board at GenSpace. We have, safety training procedures. We have a very good relationship with our local Homeland Security people. And even got together with people to hammer together a joint code of ethics. This was something that happened in 2011, funded by the Wilson Center. And as you can see, there was a code of ethics in Europe and in the United States. You guys are very safe. You can see that the European labs have more things on their code of ethics than ours, notably modesty, which I thought was very interesting. And actually, the German contingent argued very hard for personal responsibility. And they're actually embedded in a design lab. That they didn't do that. So what can you do in one of these labs? Well, I think it's this technology. Who's ever done 23andMe, something like that? Okay, so it's becoming a consumer product now, all right? It's filtering down. So 23andMe is basically finding out about yourself. You can also find out about the world around you. So these are a couple of high school students who decided to find out what was in their food. And you can do this through DNA barcoding. And so they found out that the sushi that their local restaurant was selling was not the fish that it was advertised as. And they went on to test goat cheese and all sorts of other stuff. And you don't want to know how much the food wasn't what it was supposed to be. So we had teams of students, high school students in our lab last summer doing this, everything from hot dogs to fish sticks, and it was a great time. The other thing is you can do environmental studies. There are people that are looking at microbes in the Gowanus Canal in Brooklyn. And this is in Alaska. We did a barcoding study that hopefully will be of value to ecologists looking at vanishing plant species. There was also a German journalist, Sascha Karberg, who did a DIY forensics on, to find out whose dog was leaving little presents in his neighborhood. He was actually able to identify the culprit and confront the owner. So that's useful. We also have a really good relationship with the arts community. We're a resource for them too. Narit Barshai is doing incredible experiments with sound and bacteria and fractal patterns. A little bit later, you'll hear of an architect who came to us because he wanted new materials, and we were able to start the process to engineer him some for this innovative chair. Also, we've got an artist named Heather Dewey Hagborg, and what she's doing is she's collecting hair from public places, and the technology is so powerful now. She can look into some ancestry markers and some physical characteristic markers, and she makes a portrait of somebody she's never met. I just think this stuff is really fun. That's why I'm in it. I love science. I want everybody to do it. And I would like to now say that the hands-on part of it is the best part. And I'd like to introduce you to my coworker and colleague, Dr. Oliver Medvedik, who will tell you what you can do to experience this at DLD. Oliver?
Thanks for that introduction, Ellen. So DLD organizers have been really gracious enough to invite us here and allow us to build an actual biohacker lab downstairs in the heart of DLD. So most of you here will be able to get a voucher and participate in this workshop and actually carry out some DIY bio experiments. So the workshop that you'll all be able to participate in is called DIY genetic testing. So what that means is you'll be able to analyze a teeny tiny portion of your genome, only 32 letters out of 3.1 billion letters. So it's a very tiny portion, but this tiny portion is very significant. It's going to play a major impact in curing a dreaded disease, and this disease is HIV. So it turns out that individuals who have this deletion, this mutation of just 32 letters in this receptor called CCR5, if they're homozygous, have 2 copies, they're completely resistant to HIV-1 infection. And companies now are developing drugs to essentially cure people with HIV infections based on this information. So using the technology that's available, you're able to do this type of assay on yourself. Basically, look at your own genome right here at DLD. So the way HIV gets into cells, just a little bit of background. So the virus docks with cells. It literally binds to a little receptor that acts as a doorknob. So it's a gateway. So it hijacks this doorknob to get into the cell. And once it gets in, it's able to do all its nefarious tasks. So those cells that lack that CCR5 receptor, essentially the virus just bounces right off the cells. It can't get in. So this has been some amazing information, and scientists have been utilizing it to the point that they've actually cured some individuals with HIV. So if we actually, another amazing finding is if we look at the population of people throughout Europe, 1% of the individuals have a natural mutation in CCR5 that makes them completely resistant to HIV, and about 10 to 15% have one bad copy and one good copy, in this case normal, that allows them to basically delay the onset of AIDS progression. So we're basically going to do an assay where we'll take a little bit of your DNA and amplify it, make copies of it, and then you'll be able to visualize it and check out and see what type of mutation you have. So the process is pretty simple. It's very painless. If you want to participate, you're going to scrub the inside of your cheek with a Q-tip and just hand over your DNA. And then we're going to put it in a special reagent that protects your DNA from damage. And then we're going to boil the sample, and that unlocks the DNA. It releases it into solution. So that DNA is released, but it's only a very, very small amount of DNA, so we need to make many, many copies, millions of copies of this DNA. So we're going to use an enabling technology that's gotten cheaper over the years. It's called polymerase chain reaction. And this technology, you can buy these machines now on the internet for a few $100s. They used to cost tens of thousands of dollars. And basically make as many copies of any type of sequence that you like and analyze it. So once we've made the copies of DNA, we need to see the DNA, because DNA is invisible. It's floating around in a tube. You can't see it with your naked eye, so we need to add in a chemical that makes it glow or fluoresce. And then we need to separate it out from all the other millions of pieces of DNA that are in there. So we're going to use a technology that's called gel electrophoresis. So that basically separates the DNA based on size. So remember I said that that mutation is missing a little piece of DNA. So you'll be able to determine whether or not you have this mutation using this technique called gel electrophoresis. And once you separate out the bands, you'll be able to see a size difference. So how is it going to look like? Basically, this gel here is an actual gel from one of our workshops. And on the left, you see control samples, so CCR5 on the left, then delta 32. You see delta 32 a little lower, so it's a little smaller. And all the participants you see here basically are homozygous normal. So they have 2 copies that are CCR5. I'm actually, and that's known as wild type. So I'm a wild type sample. So, okay, so you've done this workshop, you've unlocked your inner molecular biologist, you've found out what kind of mutation you have. What you can do next is, if you like, we've made some buttons, you can share this information with the participants. If you're wild type like myself, you can put on a button that says wild type. If you're a rare heterozygote mutant, 10 to 15% of you have this mutation here in the audience, you can put on this button. And if you're the very rare, ultra unique mutant, full-on mutant, we have this button available to you. So if you'd like to take this trip to the future of genetic testing and health with me, I encourage you to essentially go down to the participant booth, and we have these special tickets and vouchers that you can then take over to our booth and be able to participate in this workshop. Thank you very much.